The metabolism of the ketone body can be viewed from a carbohydrate-restricted perspective. That is, during period of prolonged fasting or dietary carbohydrate restriction, our body metabolism gradually gears toward an increased utilization of lipid resources to generate energy. Along with a lower concentration of insulin and other hormonal responses, there is a higher rate of lipolysis and increased delivery of fatty acids to the liver that ultimately lead to the formation of ketone bodies. Generally, ketone bodies production indicates that the lipid metabolism in our body has been accelerated, as well as all the enzymes that involved in the metabolic pathways of lipid metabolism. The process of ketone bodies production is termed ketogenesis and is regulated heavily on the extent of triglyceride hydrolysis in adipocytes, the transport of free fatty acids across the inner mitochondrial membrane, and the utilization of acetyl coenzyme A in the ketogenic pathway. It is worthy to note that in the abundance of carbohydrate, insulin release is increased. The increase in circulating insulin then influences many of the factors that regulate ketogenesis. For example, with its general anabolic functions, insulin tends to inhibit the breakdown of fat while promoting fat storage, therefore limiting lipid metabolism and the production of ketone bodies. In short, limiting dietary carbohydrate and prolonged fasting upregulate the body's capacity to increase lipid metabolism and produce ketone bodies. These ketone bodies, once generated in the liver, undergo oxidation, which is a process called ketolysis. The oxidation of ketone bodies occurs in many tissues outside the liver, such as the muscles, heart, and brain, where they are then utilized for energy.